complete exon or intron structures within the genes in genomic DNA. So wherever your exons, wherever your coding, so there are two options. Either you can have only the protein coding regions, or you have the just the coding regions. You can select for it. Okay, so it gives a very good scanning technique, and almost 75 to 80% of exons are identified exactly using these gene scan algorithms. So it is based on your hidden Markov models. Okay, so hidden Markov models are based on probabilities. Okay, so what is the probability of finding a particular nucleotide at one position if it is conserved? So higher the probability, if the same sequence is present at the same position in this given sequence also, then you can say yes, it's a part of your signal. So then using that, you can find out what is the likelihood that a gene is present. Okay, so that's how your signals are going to be scanned. So it uses a training data set. Now, when you say a training data set, it is nothing but already available knowledge. So हमें जो पता है already the genes which we already know. Okay, so what are the upstream sequences which are present in these genes? Okay, so that's been studied. So you have a lot of data regarding that. So that data is fed in this algorithm. Okay, so this is called as your training sets. Okay, so training sets are important for any kind of your prediction tool. Prediction ऐसे ही नहीं होता है जो हम पहले के जो भी occurrences है, when we study those occurrences, we try to find out these probabilities and all. So those earlier data is definitely important, for estimations or for predictions. So that is what is going to be done in case of your this gene scan method also. So biological input, that means you have to give the input that the sequence. So it can be codon bias in coding regions. Okay, so wherever your coding regions are there, so in that coding regions always you will be having codons. Yes, there are sequences, codons, there are selected codons. So always if it is a coding region, you will be having the sequences which are coding for these codons, right? So that probability you have to check. Gene structure, so it should have exon regions, it will have intron regions, it should have start and stop codons, then promoter regions should be there, then genes on both strands should be there. So all these things have to be, so these are your biological inputs, these are your knowledge that you are getting from your earlier known genes. Okay, so ये सब चीजें is going to be considered when you are talking about or for prediction. So it covers cases with input sequence contains no gene, partial gene, complete gene, or multiple genes. So all these things, it can happen that one particular DNA has more than one gene also. So that also will be found out using this particular method. So what are the limitations is, yes, it is an ab initio method. So hence it will not use any similarity search to predict genes. And if there are some alternative splicing and all those things you will not get. And sometimes it can combine two exons together from consecutive genes together. So when you get the output, this is how your output in your gene scan will look like. Okay. So I hope everyone is having. So for example, this is a particular predicted peptide sequence. So there are two things. Either it can give you the coding sequences. I will show you that also. So coding sequences दे सकता है या फिर predicted peptide sequence can be given by you by this particular algorithm or by this particular tool. So when it is giving, it will have this particular data also. So everything. So here there are many things that are being given. So let us think what are these. So this is the explanation about the output. So X gene X, so that is the gene number, the exon number. So ये जो दिख रहा है, so that is the gene number 1, and these are your exon numbers that's been given. Okay, and then you have type, which type of that exon it is. So if it is int, it is your initial exon. Then INTR, that is, it is an internal exon. So like it is flanked with, if it is an internal exon, it would be flanked with introns on both these sides. Terminal exon matlab last mein hoga, so it has to be like it would be after that exon you will be finding the stop codons. Then single exon gene, so there are no introns at all. So एक ही exon में initial ATG भी रहेगा और last में you will have your stop codon also. Or if it is a promoter region, or if it is a poly A site, so all these things can be given. So here you can see you will have the initial exon also, you'll have the terminal exon also, you'll have poly A regions also, from where these regions are starting, beginning, and where it is ending. So region, so if it is जो तुम्हारा length of sequences है, तो उसमें से 1884 to 2074, these nucleotides are giving you the initial exons. Similarly, you have your 3217 to 3362, that is talking about your terminal exons. So वैसा it will give you all the information. Then S. S is talking about which is the strand, whether it is actually the strand responsible which will undergo the translation or transcription, because you know only one of the strand is coded and the other strand is not going to be coded. So whether it is the reverse strand or whether it is the input strand. So reverse strand or your opposite strand. So minus रहेगा so it is your opposite strand. Plus sign रहेगा so it is your input strand. So here you are having your plus strand. Then so beginning and ending, that I have already told you. Length. So this is, we'll talk about what is the length of the sequence. Then rest of the things are not so important. FR will talk about the reading frame. So from where the reading frame starts. Okay, so whether it is starting from X, so X matlab 2, so it is starting from the second nucleotide. Okay, so from that the reading frame has to be considered. Then net phase of the exon. Then IC, that is initial signal, or your splice score, that will talk about ICE, DOT, coding region score. So these scores which are there, okay, these scores which are there, that will talk about the quality of the prediction. Okay, जो तुम्हारा prediction है, whether those predictions are proper or not. So P will talk about the probability of the exon. You know that probability should be like, if it is 1, that means it is the best probability. So you are not going to get 1 anyway because it is prediction. But this is showing you 0.98, which is very much closer to 1. So still it's going to be called as your good score. Okay. So the total score of this prediction will depend upon these scores which are generated. So these are statistical methods. Okay. There are a lot of calculations which are involved in calculating this part. Okay. So higher the score, higher is, you can say, you can be more reliable on that particular result. Okay. So the scores of a predicted feature, so whatever we are talking about is nothing but your log odds measure of the quality of the feature present on the local sequence. So a predicted 5' splice site score, so which is like very close to 100 is stronger. 50 to 100, that means it is a moderate score. 0 to 50 is a weak score, and below 0 is poor score. That means actually there are no real sites, or the prediction is very poor. That's how you can talk about. So the probability of a predicted exon is the estimated probability under GeneScan model. So which will tell you that whether the exon is correct or not. So it will talk about, it will be dependent upon the local sequence properties. That means whether it fits, how well the exon fits with the neighboring exons. Okay, so whether it is proper or not, and shows the predicted exons with higher probabilities are more likely to be correct than those with the lower probabilities. As I already told you, ये p value जो होता है, this p value should be higher. Okay, so the maximum is 1. So it will be closer to 1 are your good values that you can estimate. Yes, students, have you understood how it can be done, the gene scan thing, the output, how it is there? What I'll do is I'll first go for your gene scan method. I will be showing you the gene scan, and then I will just quickly discuss about the glimmer and the grain method. So that we are not going to perform, but just for your knowledge, we will be discussing. So I'll just switch off here. Let me go to the tab. So what you can put is gene scan. Okay, let me just show you the tab. Okay, I told you that it's, can you see the tab, students?
Yes, ma'am.
Okay, so I told you that it's present now at the MIT University ka website. So that's the website that you can see, MIT.edu. So you can find out, and so GeneScan would be present in this HTTP hollywood.mit.edu education. So you can just click on this first one. So this is the home page. Everyone can see the home page, right?
Yes, ma'am.
Okay. So let me find out one sequence. Okay. So let me go to NCBI and find out a particular. So I'll go for the nucleotide sequence. Say, let me find out CMYC gene. Okay. Let me have this uh, Xenopus lavis sequence. Okay. So I'll just copy this sequence, the entire sequence. Okay. And go back to the gene scan one. So yes, it's a vertebrate. So I'll go for that and then I'll just paste it. Okay. So here you have something called as your suboptimal exon cutoffs. Okay. So this cutoff value which is there. So if you just go for your, when you check all this, so cutoff values is the threshold value. Okay. So that means how statistically important your value is going to be there. Okay. So cutoff value should be always more than 0.1. Okay. Higher it is, best it is. So one rakhna matlab best cutoff that you are going to get. But sometimes when you are keeping it one, you may not get the result. Okay. Please understand that when you are keeping it one, you may not get the result. If you're getting the result with one, that means it is the best scan that you can do it. Okay. अगर उसमें नहीं मिलता, अगर वन रख के नहीं मिलता, तो then what you have to do is you have to change it to 0.5.
Ma'am. Oh, sorry, I was muted. Oh,
कहाँ से नहीं सुना है तुम लोगों ने? I'll go back then. Okay. So I was talking about, if you have seen that I've entered, यहाँ पे probability cutoff has to be there. So we saw at probability cutoff 1. Just excuse me for 1 second. Huh? So sorry. So you can see I have checked with cutoff 1, but did I find any exons? No. Okay, so now I am going to lower the cutoff value. And here, as I said, you have to print options में predicted CDS and your peptides दोनों भी होना चाहिए. And then again, I will run the gene scan. Okay, so here see I have got the things. So these are your predicted peptide sequence. There is no predicted peptide. These are your predicted peptide sequence 1. This is your predicted CDS, that is your coding sequences. Okay, you can see this is your coding sequence and this is your predicted peptide sequence. So peptide sequence is 419 and coding sequence is definitely it has to be multiplied by 3, right? Usi ka, this is your predicted coding region. So all the data, so जो हम लोग ने अभी discuss किया, you can see over here. So it will talk about, so this is your different that signals is your thing, and it will talk about where it is beginning, where it is ending. Okay, so here when you are talking about your scores, okay, जो total score है, so here you can see these scores are very much good, like it is more than 50, so that means it's a moderate one. Okay, not very much closer to 100, but it is more than 50. 59, 60, so more than 50, and hence it is moderate one. Wherever it is 0 या फिर lower than 0, that means you cannot be very much dependent upon that particular values. Okay, so you are not going to be dependent upon those values. So this is these are talking about your suboptimal exons. और जो स्कोर तुम्हें अभी मिला है, ये जो दिया है, whatever your predicted values that you are getting, ये predicted peptide जो है, उसका score is this. Okay, so they are very much sure about this particular values. So जो जिसका भी score high होता है, ये 65.49 or 66. Okay, so only that they are giving as your predicted peptide and your predicted CDS. Yes, students, is it clear?
Yes.
Okay, so this is how this is your results, and this is how you can find out where is your gene region and what is the peptide that is going to be there. Okay, understood everyone? So this is how your gene scan could be used. I'll just stop this.